Hi. Now that we had to postpone the CSBC conference to next summer, we are releasing short introductory videos about next year's speeches. In this video, Jarkko Rautio, a professor of pharmaceutical chemistry and vice head of School of Pharmacy, University of Eastern Finland, is introducing his research on prodrugs.
Hello everyone. The global crisis occurring due to COVID-19 has forced many scientific conferences scheduled for this year to be postponed. This also happened to Chemical Sciences in Biological Challenges conference that was originally planned to be held in June this year. Understandable, there is now the urgent need to identify effective therapeutics against COVID-19. One of the first candidates moved into the clinic as a possible treatment is Gilead's remdesivir that was originally identified during the Ebola outbreak that started in 2013. However, that compound failed in the clinic then. But like I said, remdesivir is now being tested in clinic against COVID-19. It is a nucleotide analog that inhibits viral RNA polymerases, blocking RNA synthesis, and therefore it inhibits coronavirus replication. In fact, the FDA has already issued an emergency use authorization for remdesivir for COVID-19. But I think we all are really waiting for the more concrete evidence of its benefits. So, remdesivir, shown here, is lipophilic enough to get into the cells, where it is metabolized into the nucleoside monophosphate form via a sequence of steps. Here are two alternative metabolic routes depending on the group which is cleaved first, and of course these routes can take place parallel. Then the 4-nucleoside monophosphate here undergoes further phosphorylation events inside the cells, yielding the active nucleoside triphosphate analog shown here. This all means that remdesivir is a prodrug. It is pharmacologically inactive as such, and it must undergo bioconversion or conversion into the active form in the body. That definition is good to remember. So a prodrug is inactive or significantly less active than its parent drug. Then this inactive prodrug form must undergo conversion, which in most of the cases is enzymatic conversion, to release the active parent drug. So the basic product idea here is to get a nucleoside monophosphate form into the body and there inside the cells. However, this monophosphate form gets ionized in the body, and it is too hydrophilic to allow oral absorption. Therefore, its phosphate group needs to be neutralized by lipophilic groups, which in prodrug terminology are called promoieties. So these promoieties are shown in purple colors in this figure. Then, after oral absorption, the promoieties are cleaved by various endogenous enzymes. So the approach used with remdesivir is actually pretty successful, because similar approach where the phosphate or phosphonate group has been masked with so-called phosphoramidate strategy has already been successfully applied to two FDA-approved antiviral drugs that are sofosbuvir and tenofovir alafenamide, shown here. So these previous examples represented prodrugs that were developed to overcome poor lipophilicity, which usually also means poor permeation across the biological membranes. In other words, poor absorption. Other advantages that prodrugs can offer are, for example, improved solubility to solve formulation and administration issues for poorly soluble parenteral drugs. In similar way, poorly soluble oral drugs can also be turned into more soluble prodrugs that have better dissolution in the GI tract and then better oral absorption. Then there are also, for example, several various cases where prodrugs have been able to reduce or even avoid toxicity issues by being better targeted in the site of drug action compared to their parent drugs. Here are some product numbers. It was quite difficult to find out the exact amount of marketed prodrugs, but I estimated based on the literature and recent product approvals that about 10% of all approved drugs can be classified as prodrugs. And I'm pretty sure that many find this number surprisingly high. In the past 10 years, over 30 prodrugs have been approved by the FDA. The approved prodrugs are listed here. No need to read them aloud. At least one product was approved last year. So the use of prodrugs can be thought as a problem-solving strategy, but as exemplified by those lipophilic phosphoramidate prodrugs, when this strategy is planned carefully, it can definitely be very effective in making better drugs. And what clearly has changed in the past years is that prodrugs are no longer seen as the last resort for poor medicinal chemistry, but teams are willing to use prodrugs more often when problems arise, and in that sense, prodrug strategy is now seen an integral part of the drug design and discovery strategy. I hope I now have awakened your interest in prodrugs. I'm Jarkko Rautio, Professor of Pharmaceutical Chemistry and Vice Head of the School of Pharmacy at the University of Eastern Finland. I have 20 plus years of experience with prodrugs, and I will give more detailed talk about the topic in the Chemical Sciences in Biological Challenges conference in Kuopio 2021. So I hope I will see you there.